Hey everybody, how are you? What we're gonna do now is Mendelian inheritance or trait inheritance. So it's, of course, continuation basically of genetics, molecular biology, but the difference is in trait inheritance, we're not really talking about the molecular aspect. That is not the A's, T's, C's, and G's, not the enzymes, not what's going on at the genetic level, but what we actually see when two organisms mate with each other, breed with each other, fertilize each other, and then pass traits along. So this is the big question for Mendel was, without any kind of biochemical tests, because we didn't know what DNA was back then, we didn't have genetic testing, we didn't understand that DNA was the genetic material. None of this had been discovered. So really all Mendel had to go on was looking at two plants, crossing them with each other, and then seeing what the offspring looked like. Now, the reason for this, if we want to talk about the historical background, which is one of the learning objectives here, is that the 1800s was a time of tremendous social change. Science was coming around in many fields, particularly hygiene. Some medical advances had been made, vaccinations. More and more people were surviving childbirth. And what this means is population growth. At the same time, we had an industrial revolution going on. So many people were moving into cities where the factories were being built and away from the farmland. So you had more people and thus need for more food, but fewer people on the land to provide the food for the people in the cities. So what do you do about that? If you're a good ruler, a good leader, you understand, at least from a selfish point, that a hungry population is a dangerous population. When people are hungry and starving, you're more likely to have revolutions. So, the governments were aware of this, and they were looking for ways to feed their people. I don't know if this directly translated into Mendel's work, but this was part of the historical background. So Mendel decided to look at how traits are passed from parent to offspring in a crop, a common crop, the lowly garden pea. And he figured that if he could understand these pea plants, how they pass traits along, then that understanding of pea plant trait inheritance could be used in other crops. Now, he chose, and this is the next big thing. So that's the historical background, right? We understand the Industrial Revolution, increased lifespan, increased survival, increased populations, and the need to feed the people. That's what's going on historically. Now, Mendel chose the pea plant. The next big thing I want you to understand is, why did Mendel choose the pea plant? Well, it wasn't random, and it wasn't because it's the only thing he had available. I think if we look at his experiments, we can understand, certainly surmise, that he had very good reason to choose the pea plant. And this brings us to a couple terms that I want you to be familiar with. The first one is an experimental model. Now, what do we mean when we say experimental model? Okay, Mendel is using the pea as a model for trait inheritance in crops. So we're not really particularly singularly interested in the pea plant. Right? We're hoping and assuming that if we use this as a model, we can apply what we learn to other plants and increase crop yields, productivity, and other things in addition to the pea, right? And we know that we use experimental models, that mice, rats, rabbits, in the lab, not because we really care about extending the longevity of a mouse, but because we hope and we assume that what we learn from the mouse we can apply to human medicine, because we're physiologically similar, right? Okay, so that's your model. And of course, when you're choosing an experimental model, there are several factors that come into why did you choose the pea plant? And I think if we just thought experiment, why was the pea plant a good model for Mendel? You can ask yourself, well, why didn't he choose elephants, right? What's the difference in trying to do research on a pea plant and an elephant. And there are some really good reasons, some very obvious reasons, and some not so obvious reasons. The first is, obviously, peas are pretty cheap, pretty easy to grow, and you can control them fairly well, right? I mean, they're not going to run off on you. They're not going to, charge the gate and try to escape. You can also selectively breed them, right? Take the pollen from one, put it on the flower of another. I'm controlling which organism is breeding with which other organism. Whereas you can kind of imagine trying to control a big bull elephant when it's chosen to mate, and you try to convince it that it shouldn't mate with that other elephant. It might be a little more difficult than controlling the pollen from one pea to another. The other thing is that elephants are massive creatures. They require a lot of food, and their generation time, and this is not gestation time, they take about 2 years to develop an elephant baby in the mother's womb, but about 18 years to where they grow up to be old enough to breed. So you're talking, for one generation of elephant data, you'd have to wait human generation, 20 years. With peas, you can breed 4 times a year. You can breed massive numbers of plants. So it's just not, it's clearly on that kind of controlling the plant costs, ease of using the pea plant. It's pretty obvious why you would choose the pea over some other model. And I'm going to pull this up now so we can see. Let's see what we got here. Screen share. Right. So if we're down here now, we'll come back to this now. We'll talk about that in a minute. But first of all, peas are cheap and easy to grow, right? So that says it all. Cheap and easy to grow, and also easy to control who's mating with who. Peas have a fast generation time. Like I said, you can grow multiple crops a year as opposed to waiting a generation, 20 years for one baby elephant to grow into a reproducing organism. All right, now that's pretty obvious, right? Those two, pretty quick and easy. That's no problem. But this is a little more, also very important. Now, what does this sentence mean? Peas have a variety of variable phenotypes. Well, if all peas were all the same all the time, it would be hard to tell if there's any, which trait is being passed from which parent. So the point here is that if you look at a pea plant, this particular species of pea, if we can measure height, and we can easily measure height by looking, when you can use a ruler if you want to be more objective and get actual numbers. So quantitative measurement, you can measure the height with a ruler. And the thing is that the peas are either tall or they're short. There is no in between. So there's no confusion. Did this baby pea plant, did it get the tall gene or the short gene? You can tell just by looking, right? Another one, the flower color. Okay, purple or white. It's obvious when the plant sprouts its little flowers, they're either purple or white, so you can measure it. So really what Mendel's doing here is kind of a colorimetric test, right? And we talked about colorimetric tests in the lab. You can easily discern the difference, right? Okay, not only flower color, but pea color. Are they yellow or green? Okay, maybe a little tougher if you're colorblind, but nonetheless, you could look and see if the peas were yellow or green. And there were others, like, was the seed pod vacuum packed, was really tight around the peas, or was it loose? So things that there was no confusing, right? It's tall or short, purple or white, yellow or green, tightly packed or loosely packed. And this allowed him a way to measure which genes were being inherited. And that's very important in this experiment. It shows that he understood he needed a way to be able to measure and tell which genes were being passed. And this one, even more important to understand. If you want to understand his experiments, we have to look at this last line here and say, okay, peas can either self-fertilize or cross-fertilize. And what's the importance of that? Well, if you self-fertilize a plant, then whatever you see in the offspring, you know where those genes came from, right? It must have come from the only single parent that produced that plant. And this allows you to avoid the confusion of, well, this little baby plant has this characteristic, a purple flower, a white flower, it's tall or short, and asking, well, did it come from the mother or the father, the male or the female? And that's another thing we should know, is that plants, we do consider them to have male or female characteristics, right? We won't go any more into that, but they do have kind of gender. Some plants are all male and then all female, but on some plants, the single plant growing there itself has both male and female characteristics. It produces pollen, and then it has whatever female I think stamen, something like that, to receive the pollen and fertilize a little ovum in the plant. So that's very important. Male and female, but you can either cross-fertilize, take this plant with this plant and cross-fertilize them, or a single plant that fertilizes itself, and then you know in the offspring where those genes came from, the parent. For instance, if I have a tall plant and I self-fertilize, well, I can assume that the offspring would be tall because that's the trait of the parent. But what if I see a short plant? That's kind of weird, huh? How did that happen? And this ends up actually being very critical, very important in what Mendel did. And almost addresses one of the main questions is that Mendel was dealing with is, can a plant that shows one phenotype, and we'll discuss that word in a minute, one physical characteristic, such as being tall, can that tall plant pass on the other trait? And in the end, we're going to see that Mendel found, yes, indeed, you could do that. And this tells you something very important about the genome of eukaryotic organisms, something that we've already covered. If I know that a tall plant must have the tall allele because it is tall and it's expressing that gene, right, that gene variant, that allele, but then I learn it can actually pass on short information to its offspring. That tells you that it must have two versions, right? It must have a tall variant of the gene for height and the short variant for the gene for height. Now, what does that mean? That means that the eukaryotic cells are diploid. We talked about that in one of the last previous chapters. And this is pretty amazing considering that, Mendel uses no molecular techniques. He's not looking under a microscope. He's not tracking the chromosomes. He doesn't understand anything about DNA. But nonetheless, he's able to determine with these experiments that our eukaryotic cells are in fact diploid. Now, what else he finds out when he does that is that each plant passes on only one or the other variants that it might have, one or the other alleles, meaning that if I have a tall plant carrying the short allele, when it produces its gametes, and we talked about this, that the gamete is only carrying one version. It's going to pass on either the tall or the short. Now, what does that mean? That means that your haploid, your gametes, your sperm and your egg, are in fact haploid. So again, a really fundamental thing about how humans and other eukaryotic cells reproduce, how we pass on genes, discovered by Mendel without ever knowing what a gene was. In fact, he's the one who developed the idea of genes. He didn't call them genes. He called them, I think, heritable factors or something like that. Of course, he was German-speaking Austrian monk, so he called it something else. Now, what do we call that if I have, let's take this off for a minute. If I have a go bath, and the way we did this was we said that, we have a maternal and a paternal chromosome, and I've got red hair and black hair. But when I make my gametes, my sperm and my egg, I'm only going to pass on one of these. The idea, if we take these homologous pairs, once again, right, this is chromosome number 1, this is maternal, this is paternal, these are your homologous pairs, that when I go through mitosis, then the cell is going to get, both of these, the maternal and the paternal. But if I go through, so this is this is a review from a previous chapter we talked about, right? Eukaryotic cell replication. If I go through meiosis, then I end up with, either the maternal or, so that's the little tail, right? The black hair. The sperm and egg only have one of these. What is this idea called? That when I produce my gametes, my sperm or my egg, they only get one or the other of the alleles. This is called gene segregation. And that should be pretty obvious what that word means if you think about it, right? Segregation, keeping things separate. We're separating this allele and this allele in the gametes. Okay, so an important term. Aha. What else? Let's go back to the study guide and we can walk through the next important stuff and rehash what we just talked about. Now, I will ask you about the important reasons for Mendel's choosing the pea plant. And you should know that these are the reasons, right? They're cheap and easy to grow. They have fast generation time, variable phenotypes, right? For each of these phenotypes, height, flower color, pea color, and others, that the plants had variables in those, right? As far as height goes, you could be tall or short. As far as flower color goes, you could be purple or white. As far as pea color goes, you could be yellow or green. Now, one thing that they often do in this, and if you had high school biology, assuming you did, they probably made you memorize all of these, which one was dominant, which one was recessive. Don't worry about that with me. I'm not going to ask you that. This is something that just clogs up the overall comprehension of the ideas. And these ideas are more important than you spending a lot of energy memorizing the detailed facts. If you go into plant genetics, you will have plenty of time in the future to learn all the details that you need to have. But generally speaking, cheap and easy to grow, fast generation time, a variety of phenotypes that are easily observable. And that's pretty important, right? Because he didn't have any biochemistry, he didn't have any molecular biology. And then very, 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 very important, peas can self-fertilize or cross-fertilize. Okay, there we go. Now, we're going to need a couple terms, about 4 terms, really, in order to discuss what we're seeing in Mendel's experiment. And we need to talk about genotype and phenotype. So what is the genotype? The genotype is the, can you see that? A genotype describes the genetic makeup of the organism. So I know that if I have on chromosome number 1, my maternal and my paternal copy of the chromosome, if they're both the same, right? So they're both red hair alleles, then these are the same, right? So we have homozygous. But this word is a little incomplete. When I'm describing the genotype, well, it could be, if it's homozygous, all I know is that they're both the same, but we can ask ourselves the question, which one are they? Is it homozygous red or homozygous black? Now, again, without getting into many details, right, we're going to say that when you're talking about homozygous, we have to say, is it homozygous dominant? And we'll discuss what dominance means in a minute. Or is it homozygous recessive. Without this qualifier here, if I only say that the genotype is homozygous, then that just tells me they're both the same. It doesn't tell me which ones they are. So any kind of answer about a genotype, if you don't see a combination of these, either homozygous, recessive, or dominant, then you have to exclude that option because it's not a complete answer, right? So any discussion of genotype requires homozygous and dominant or recessive. Or the other option is heterozygous. Now, this is a little bit different because if I say heterozygous, what does that mean? That means I have one of each, right? I have one red and one black. Now, it doesn't make any sense to say heterozygous dominant, right? Because it's not heterozygous dominant, it's heterozygous. We have one of each. So this word suffices, is adequate to describe this situation, one of each, right? This word alone is not adequate to describe it. We have to tell you if it's the homozygous recessive or the homozygous dominant. All right, just keep that in mind when you're thinking about, we're gonna have to describe the genotypes of what Mendel sees in each of his generations after he runs through, mating plants and developing true breeding strains and everything he does. But when we look at the offspring, we're going to have to say, okay, well, what do they look like? And what's the genetic makeup? What's the genotype of them? So keep this in mind, homozygous dominant or homozygous recessive, or simply heterozygous, depending on what they have. Okay, now, the genetics is one thing, and the other term we want to think about is not the genotype, but the phenotype. Now, what is the phenotype? The phenotype is a description of the physical characteristics, the observable physical trait that you see. A physical description, right? So is it tall or is it short? Are the flowers purple or white? Are the peas yellow or green? And this is based just on what you see when you look at the plant. And this is going to be important because when we do our cross using the different pea plants, if I take a tall plant and breed it with a short plant, what do I see from that offspring? Okay, that's the phenotype. And that's not going to involve any homozygous or heterozygous because that refers to the alleles. What that's going to be is either, and for our purposes, we're just going to say it's either dominant or recessive. Any question on the test about phenotype, asking you what phenotype, is simply going to be dominant or recessive, okay? Once again, I'm not going to ask you if the tall was dominant or recessive. We're not even going to use those in the questions. I'm going to ask you what happens if I cross a homozygous dominant genotype with a homozygous recessive genotype. What was the phenotype that I see? And you don't have to say dominant or recessive. And this, we'll figure out how to determine that as we go through the rest of this lesson. All right, so we got a couple words, genotype and phenotype, and you need to know what terms we use to describe those phenotypes. Those terms are, once again, review. Regarding genotype, you can be heterozygous dominant or homozygous dominant or homozygous recessive or heterozygous. Those are the only options, right? You must include for homozygous dominant recessive. However, when we talk about phenotype, the only possible answers are it's either dominant or recessive. Now, what does this even mean? Well, we're talking about crossing plants, and let's say I take a tall plant, short plant, and let's talk about this now. We're not going to write out tall and short every time. If I have tall versus, well, let's proceed with this. Now, depending on the genotype of these, say I have a homozygous tall, and a homozygous short, and I cross them with each other, what do I get? I get tall all the time. Now, what that tells me is that the tall trait is the dominant trait. Mendel discovered this. He came up with the word dominant because he did exactly this. He crossed homozygous dominant, homozygous short plants, and he saw that only one trait came up all the time in such a cross, and he called that one the dominant because it's the one that dominates. If you're mixing tall with short, homozygous tall with homozygous short, then you always see tall. And what we do is we take this letter and we use it for the dominant to suggest dominant. So I can simply put a T here since it's the first letter of the dominant trait, and that means dominant, capital T. Now, what do I do for this one? I don't use an S. What I do is I use a lowercase t, and that tells me you have the recessive trait for height. And this is a little complicated. Why don't we use an H? Who knows? Why don't we use a T and an S? It's just easier. Whenever you see a capital letter, that tells you that's the dominant allele, the first letter of the dominant allele. When you see the lowercase letter, that's the recessive allele, even if it doesn't match with the first letter of this, right? We always go back. In both cases, we're coming back to the T in the dominant trait. This is the way it works. So if I tell you that purple is the dominant trait, white is recessive, what would our shorthand for that be? Well, obviously P, purple, right? This would mean dominant purple, and this is going to be the white plant. Dominant, recessive. Okay. Okay, so the next thing, if we step back a little bit, we think that I said that one of the questions, or maybe the question that Mendel was wanting to figure out was, if a plant has a particular trait, such as being tall, can it pass on the short trait to its offspring? That was kind of the big question, because if you want to grow tall plants, which you probably do, because you get more productivity to feed all those people in the cities with fewer people on the farmland, historical context, then it's important to know that if I have a tall plant, that I'm only going to get tall plants out of it, right? We don't want to breed a whole bunch of tall plants and then find out that our whole crop is short plants, because you don't get as many peas off of a short plant, right? So it's kind of important to know that, hey, these are tall plants and they're only going to give you tall plants. Now, that was kind of the question, and I suspect that people already knew the answer. I suspect that Mendel already knew the answer, because we're totally familiar with the fact that, oh my God, this little baby has red hair. I don't have red hair. You don't have red hair. Where did the red hair come from? Oh, grandpa, great-grandpa had red hair. And this trait was silenced in his offspring and then silenced in you, but it shows back up. But that's anecdotal. We don't rely on that kind of stuff in science. We've got to do the experiment as always. And Mendel's experiment definitively shows us that yes, indeed, traits can hide out and be passed on, even if the parent doesn't show that trait itself. Okay, we've got the blue-eyed, brown-eyed question, right? That's always one of the big ones. Can two blue-eyed parents have a brown-eyed child? Can two brown-eyed parents have a blue-eyed child? And we're not going to get into that, but the answer is, if you're showing the dominant phenotype, then yes, you can pass on the recessive. And you don't always pass it on. You may be homozygous dominant. But the key comes in that if you are heterozygous, if you have one brown allele and one blue allele, then of course you'll only show the dominant phenotype. But due to gene segregation, when you're looking at the brown and the blue, you may end up segregating out and passing on that blue allele to one of your offspring. And if your mate also has the blue allele, if they come together, it's going to be homozygous recessive So the only option for that child is to show the recessive phenotype. So that's how that happens. It only works if the parents, it only works if when you have the dominant phenotype, you may pass on the recessive. Obviously, if you're showing the recessive phenotype, the only way that you express that recessive phenotype is if you are homozygous recessive, right? So if I look at that, I have a parent with, I'm going to use these R's for recessive, right? The only option during gene segregation, if I make my zygotes, is to pass on that recessive allele, right? So somebody showing the recessive phenotype can only pass on the recessive allele. But if I have a dominant and a recessive, well, what's this guy going to look like? He's going to show the dominant phenotype, but he might pass on the recessive allele, right? So bigger. He can pass on either one of these to his offspring during gene segregation. Mendel discovered this. It's his experiments, the ones we're going to talk about now in a minute, that showed, yes, you can be tall and pass on the short, but you can't be short and pass on the tall. Okay. I always say that Mendel kind of understood this. He knew this was the case. And the evidence for that is the way he designed his experiment. This is another thing we want to talk about is experimental design. So if the experimental model is what you're researching on, are you using a mouse or using a pea plant or using elephants, whatever, experimental design really explains exactly what you did, what the procedures were. And we can go back to the first chapter, and we covered this in the lab. How do you design an experiment? Well, you need controls, and you have to have its hypothesis, and the experiment needs to either support or reject your hypothesis. Step by step, details we've studied in the lab, scientific papers, and we know that there's a materials and methods section where you lay out everything that you did in very high detail so that anybody on the planet can try to repeat your experiments. And if somebody tries to repeat your experiments, they should get the same results, right? If they don't, then something's wrong. Either you did something wrong, they did something wrong, you didn't tell them what to do, or somebody was sloppy, right? Or somebody wasn't aware of a variation in the procedures. We can go back to Pasteur. I think we talked about Pasteur, maybe in my video, where Pasteur sterilizes some chicken broth, he heat boils it and shows that nothing grows until he adds a little bit of dust, this Swan Neck flask, and then you see germination, you see growth and putrefaction, growth of bacteria in the chicken broth. And he was very careful to show that if you heat sterilize, then you kill all the bacteria, nothing grows. Well, along comes Tyndall, he does the same experiment, and he finds that after he heat sterilizes, he does get growth. Now, this is a direct contradiction of Pasteur's results, and it was probably confusing to them at first until you know a little bit more. The reason that Tyndall got a different result is because he used a different model. He did not use chicken broth. He used what we call hay or grass infusion. Now, if you know anything about soil bacteria, that will be all over the hay or grass that you boil, is that they produce heat-resistant spores. What that means is when Tyndall heat sterilized his grass infusion, he was not sterilizing. These little survival pods, I like to call them, these little protein coats containing bacterial DNA, were able to survive. They're not really alive, but they're not growing, but they made it through the heat process. And as soon as things cooled back down, they could grow. Now, that's very important. When you do an experiment and you want to publish your results, you have to make sure that you tell people exactly what you did and We saw that in the lab. And then if you're trying to repeat and confirm somebody's results, you have to do exactly what they said. If you change something, then you may get surprising results. Now, in that case, it was actually fortunate because it taught us that for crops, heat sterilizing may not be enough to kill the bacteria. You can't just boil it. And certainly even today, you can get some serious foodborne illnesses from crops because they do carry endospores. If something goes wrong in the cooking process, if you don't cook them long enough at high enough pressure, the spores survive. You can get botulism, anthrax, all kinds of pretty nasty diseases because those spores survive. Okay, now that's a little off track. We're going to come back to where we were. Experimental design. What exactly did Mendel do? Now, I'm going to go back to the note set because that's got critical information for us. All right. The first thing he did was created true breeding strains. And we look at this word. What does that mean? True breeding. Well, that really shows that Mendel knew that there were non-true breeding strains. What does that mean? That means I could take a tall plant, self-fertilize it, and get a short plant. What true breeding means is that whenever Mendel took that particular plant and self-fertilized it, that he would only get the same phenotype as the parent. Okay. True breeding. You can compare that to non-true breeding. What that means is you take a plant, and very importantly, you self-fertilize it, so all the DNA is coming from that parent. In a non-true breeding, you may self-fertilize it and see the different trait, right? Now, what does that mean if we look back? If I'm talking about a true breeding strain, what that means to us today is he developed homozygous strains, right? Homozygous dominant or homozygous recessive. Now, at this point, he didn't know about homozygous and heterozygous. He hadn't yet discovered that there were two alleles. So he called them true breeding. We would say that those true breeding strains are, in fact, homozygous, either homozygous dominant or recessive. He didn't even know about dominance and recessive yet at this point. He was just developing his strains. Now, key terms, right? The parental generation would have been these true breeding strains that were, in fact, homozygous dominant or homozygous recessive. So now let's say on one hand, I've got some plants that are true breeding tall. They always breed tall when self-fertilized, right? And I have true breeding short. And if I self-fertilize those, then I always get short plants. That's my parental generation. Mendel's next question was, what happens if I cross them? I have a plant that always breeds, self-fertilizes tall, and I have a plant that always self-fertilizes short. What happens if I cross-fertilize those two? This is actually where Mendel discovered the idea of dominance. Because if he took true breeding tall and true breeding short, and he crossed them, he always saw the tall offspring. If he took purple and white, he always saw only one of those colors in the offspring. And this characteristic, this phenotype that he saw, he called the dominant phenotype because it beats out the other one. Okay, now let's look here, right? We cross-fertilize the parental strains having different traits, tall with short, right? He calls these, and we'll have to follow this to make sure that we know which generation we're talking about. We start with cross-fertilizing the parental generation, right? And what we get is F1. Now, why does he call it F1? This is from the word filial in French. If you speak French, fils is child, fille is girl, right? Filial means family. offspring. So the first group of offspring that he gets when he self-fertilizes the true breeding strains is the F1. And this is where they always show the same trait, right? So always tall, always purple, always yellow, whatever. Mendel names this the dominant trait. So all F1 generation from tall true breeding by short true breeding were tall, always, all the time. So that's what he calls the dominant trait. Now we're going to use a Punnett square. I'm not going to have Punnett squares on the test, but if you want to see why we get this result, knowing what we know today, if we look at a Punnett square, and Punnett squares consider gene segregation. They separate out the 2 alleles for the mother and the 2 alleles for the father, and we cross them because we know that those alleles are going to match back up with one from the mother, and the father. That's what the Punnett square is for. So I take my X here, the box, and let's do this. Let's say this is the maternal and this is the paternal. Well, what are the alleles that the mother has? Let's say the mother was the tall allele, right? So knowing what we know now, we would say that she has a homozygous dominant genotype, but only one of these is going to cross with the other parent, right? So we have these 4 boxes. For this allele, there are 2 options here from this one that it could possibly, this allele from the mother could possibly cross with, but we know that the father is true breeding short. What happens when these reunite in fertilization is that I get one big T and one little t. So in all cases, when I'm crossing these with each other, all of the offspring genotypically are heterozygous. So in the F1 generation, the genotype is all heterozygous. Again, Mendel couldn't measure this. What did Mendel actually see? Well, in a heterozygous, we're only going to see the dominant trait. So in the F1, Mendel sees nothing but dominant plants. He says, okay, they're all tall. We're going to call that dominant. And does that settle the issue? No, it doesn't. When we do this, we understand why they're all dominant phenotype is because the offspring of the F1 generation are in fact all heterozygous. Okay, now Mendel continues his experimentation. He could have hung up his cleats then and said, okay, I've discovered that only one trait is passed on, but he didn't do that. After crossing the true breeding tall, the true breeding short, he wanted to find out what was going on inside these all dominant, right? So he wanted to figure out this. The question is, okay, in the F1, what I see is all dominant. But what happens if I go back to self-fertilizing these guys, right? Sure, they're all dominant, but what's going on inside? Is this dominant plant actually carrying the hidden allele, the recessive allele, or has it been destroyed? How does he do that? He takes these and he again starts self-fertilizing them. And that way, if he self-fertilizes this plant and he sees a short plant out of that offspring, then he knows that that allele did not disappear. It didn't go away, that it's being carried and can be passed on, right? Okay, so you start self-fertilizing. The F2 generation, then, if we look at the self-fertilization, since it's the same plant, they're both going to be both parents are the same, and we know that both parents are heterozygous genotype. Now, what do we get when we segregate these alleles and cross them with the parent, which is the same parent, right? It's not a cross-fertilization, it's a self-fertilization. Well, in some cases, we're going to get this. In some cases, we'll get this. And occasionally, we'll cross these two together. And this is where we see the recessive phenotype come back. Now, once again, if we look at this, what are we going to see? This little guy is going to have the dominant phenotype because he's heterozygous. This guy certainly is going to have the dominant phenotype. And this guy is going to have the dominant phenotype. So what we see, and what Mendel saw, was a 3-to-1 ratio in this F2 generation. Again, when I ask you what Mendel saw, I'm asking you about the phenotype because he was not using molecular techniques. He was just looking with his eyes. And he was probably pleasantly surprised to say, aha, it does come back. And now what does he discover here? He discovers that the tall parent was carrying along the recessive allele because it passed it on when self-fertilized, right? Okay, so that's the F2 generation. What did Mendel see? And that means what's the phenotype? He saw 3 dominants to 1 recessive. What does he learn from this? He learns that we're actually diploid. We're carrying along this allele, even if we're showing the dominant trait, right? Because remember the F1 generation, F2 is the F1 self-fertilizing. The F1 was all tall. When I self-fertilize those, I suddenly find the recessive comes back. We're not going to go into much detail beyond that. Mendel did continue his studies, and he discovered something else very important. The question now could be, okay, so I have a tall, I have height, and I have pea color, and I have flower color. Are those passed on as individual units? If you have tall, is that gene also code for, purple flowers and yellow peas? Or is that each of that bit of information for height separate from flower color, separate from pea color? Now, what does that mean? Does a single gene control one trait, or does it control multiple traits? And what Mendel discovered was that for each trait, such as height or flower color, the genes are separate from each other. And the way he did this was really complicated. He did a double hybrid cross. We're not going to get into it, but we should understand that he did a ton more work, and he is also the person who developed the idea, kind of one gene, one, we say one gene, one protein, but one gene, one trait, right? If you have a gene for eye color, it's totally separate from the gene for your height, from the gene for your, whatever. Again, this was the dihybrid cross, and we won't go into more detail on that. Mendel got a little bit lucky with this. If the genes had been closer together, he might not have seen the same results, but he's the one with his dihybrid cross who said, okay, every trait is controlled by a separate independent gene. And that's what we assume now, right? You have a gene for eyes, you have a gene for hair color, you have a gene for whatever. All right, going back to the screen share, right? You really need to understand and be able to answer on the test, what he's doing, continuing self-fertilization. Is he self-fertilizing the one, right? What is he getting? And you need to pay attention on the test. Am I asking you what the genotype is or the phenotype? So you need to know that. What was the genotype of the true breeding strains? Well, they were either true breeding tall, right, or true breeding short, meaning they were homozygous dominant or homozygous recessive. Right? You need to understand that. about the F1 generation. You need to understand what happens when you cross them. If I cross true breeding tall with true breeding short, I'm crossing homozygous dominant with homozygous recessive, and I'm going to get all heterozygous genotype, but all dominant phenotype, right? So the genotype of the F1 generation, heterozygous, phenotype, dominant. If I then continue to self-fertilize each of those, what do I get? Oh, there's one more thing that Mendel did. If I self-fertilize the F1 generation, then what I get is, phenotypically, what did Mendel see? Phenotypically, 3 dominant, 1 recessive. And then he went ahead and he continued self-fertilizing those. Now let's see what happens when we do that. Sorry, we got off. If I take the F1 generation, which is F2 generation, which was three true breeding, 3 dominant and 1 recessive phenotype. What happens when I self-fertilize these? Well, clearly when I self-fertilize this one, I'm going to get true breeding, which equals homozygous dominant genotype, right? What did Mendel see? He saw that this guy was true breeding tall. We can say that true breeding is genotypically homozygous dominant. Now, when he takes these guys with short, he's going to get true breeding recessive. What that tells us is that both of the alleles are the same. This is homozygous recessive. Homozygous recessive. But now what about these two? What happens when he self-fertilizes these guys, right? Non-true breeding. Because since they're carrying this allele, when I self-fertilize, I get the same result as my F2 generation. You're going to have that. They're certainly appearing, their phenotype is tall because they have this dominant allele. But we know that if I cross-fertilize these guys can hook up, right? And you end up seeing that recessive trait. So these are non-true breeding. So what did Mendel find? He found a ratio of 1 true breeding, homozygous dominant, true breeding tall phenotype, homozygous dominant genotype. And then he to 2 non-true breeding or heterozygous. And then he found 1 true breeding recessive or homozygous recessive genotype, right? Okay. Yeah, it's complicated. It's tricky. There will be a couple questions about this, and I will ask you for either the genotype or the phenotype of any of those generations. You need to understand and be able to communicate using the right terms: homozygous dominant, homozygous recessive, heterozygous dominant genotypes, or dominant and recessive phenotypes. What did he get in each one? The only way you're going to get this if you go through this again and again and work it out and understand the process. Like I said, there'll be a couple questions on this in the test. And, if you just think this is way too much and you can't do it, sometimes you have to decide, well, I'll take a hit on those 2. Just won't answer them properly or trust it to luck. But I hope you don't do that. I hope you can work through it, rehearse each of the steps, asking yourself, what do you get each time, genotypically and phenotypically. Use the Punnett square to determine what arrangements you get, and then describe them with the right terminology. All right. So I believe that is about it for this chapter. No. Oh, no. A couple more very important things, right? Now, Mendel did fabulous, fantastic work, but he also got pretty lucky. He happened just accidentally to choose a model in which the genetics was really, really, really clear. If any of those alleles, any of those genes that he studied had been, on the same chromosome or closer together on the chromosome, his results might have been much different and much more confusing. So it just so happens that the pea plant has nice genes for height and pea color and flower color that are separate from each other. Because of this, and because genetics is actually much more complicated than this, I kind of say Mendel got pretty lucky that he was looking at such a simple system, such a basic system. There are only certain times when Mendelian inheritance works, and we need to know those conditions, right? In order for Mendelian inheritance to work, the case must be that each trait is controlled by a single gene. This is not always the case, right? But in the case of the pea, luckily, height is controlled by a single gene. Color is controlled by a single gene. So Mendel got lucky, right? They could have been more complex. Mendelian inheritance rules, trait inheritance rules only work when each trait is controlled by a single gene. Now, the other one, there can be only two alleles for that gene. It must be either tall or short. What happens if you have an in-between, right? What happens if you have, blue, green, hazel, and brown eyes? It's more complicated. So you can't use Mendel's rules in all cases. You can only use them when there are simply two alleles, tall or short, purple or white. And then there has to be a really clear dominant recessive relationship between the alleles. That's not always the case. We can have codominance or incomplete dominance. So again, more confounding factors that are going to, confuse your results. Now, our blood typing system, A, B, O, and AB, is based on codominance. If I am homozygous A, then I'll have type A blood. If I'm homozygous B, then I'll have type B blood. But if I'm heterozygous, if I have both A and B alleles, then I express both of those on my red blood cells, and my type blood is AB. So neither one is dominant. If I have both, if I'm heterozygous for those, then I'll express them both equally. Now, a couple other words we want you to understand, right? This word, in this case, each trait is controlled by a single gene. That doesn't apply if a trait is polygenic, right? That means multiple genes. If I have multiple genes for one trait, then this can be confusing. Incomplete dominance, we see a blending of traits and word association. Once again, when you hear the word incomplete dominance, you should automatically think a blending. An example of that, we may have a plant that has red flowers and white flowers, and when we cross-fertilize them, we see pink flowers. So that's neither white nor red. There is no dominant or recessive. It's a blending of the traits, right? Now, that's a little different, and we're not going to get into the details. I just want you to know this term now. In codominance, both traits are expressed equally. And where do we see that? Human blood type. Once again, I can be type A, I can be type B, or I can show both A and B. So that's different from pink. Red and white giving pink, right? Red and white giving pink is a blending. A and B giving AB, you're seeing both of them. You're not seeing some weird mishmash of those blood types. You're seeing the type A and the type B. Okay, now that does it for this chapter. I'm not sure how long this video is, but it's pretty, comprehensive. Make sure you're using the study guide. Make sure you really understand what's going on and you understand the terms, and that you can walk your way through and answer questions about the parental generation, about the F1 generation, about the F2 generation, and about the continuation of the self-fertilizing. Make sure you understand what Mendel did with each of those, what he saw. Were they genotype? Were they true breeding? Were they non-true breeding? Were they, would we call that dominant or recessive? Would we call it homozygous or heterozygous in each case? All right, y'all, that's it for me. Let me know if you have any questions, and see you in, I'll have one more video to give you on gene expression. All right, that's it.